Okay, I want to talk a little about morphometrics, which is a bit of a bee in my bonnet, as they used to say. I've been doing some work on it, and partly I just had to learn what these morphometric models were and figure out how you connect them to Brownian motion-y kinds of phylogeny work. And I think I finally do understand them, which is very difficult because they have their own terminology, which is very geometric and not so much evolutionary biological, even though many of the people doing it are evolutionary biologists. It's sort of they're often their own bubble with terminology, but I think the bubble is coming back in contact with everything else. My co-worker on the stuff on morphometrics is this guy, Fred Bookstein. And Fred, some of you will know him as, he's first of all Mr. Morphometrics. I mean, he's the biggest contributor to modern morphometrics. He was for many years at the University of Michigan, but some people may not be aware that a few years ago he retired, and he had had a long association with Seattle, coming to work on some facial morphology stuff with people in the medical school. And he and his wife ended up basically retiring to Seattle, except Fred is continuing research on a sort of soft money basis. He just finished also a period as professor of anthropology at the University of Vienna, mostly concerned with these morphometric issues. And we kid ourselves that we have a common ancestor who we've denoted as JFL Bookstein. If you want to know how to do that, it's very simple. You take black and white images facing forwards. You put them in a program like Adobe Photoshop or GIMP, and you make them 50% transparent, and you size them so that the eyeballs are exactly superimposed. And then you just plop them down on each other, and that's what you get. It works very well. Anyway, so that's JFL Bookstein, who represents our consensus. And modern morphometrics, which involves capturing forms either by continuous digitization, or I'll be talking mostly about landmark methods, went through a period in the 1990s when they got very excited and achieved what they describe as the morphometric consensus, which was that they reached a unified view of how to take images captured that way, forms captured that way, and analyze them in a coherent way. And I'll be describing that, but I'll also be presenting in the end what is an alternative to it. The other thing that morphometricians have started to do is that in the 90s, all the methods were treating each specimen as independent, identically distributed. Whenever they tried to analyze what changes of form there had been, there were no phylogenies around. Now, they've started doing them. The first papers on doing morphometrics on phylogenies include work by Michael Rosenberg and Dean Adams, and the earliest work is in early 2000s, and then it starts to gain a little bit. So now I estimate there's some much bigger number of papers. Here's a bunch of papers showing people doing morphometrics on phylogenies, using usually the morphometric consensus methods, which I'll give you a little introduction to in a moment. So this is just to give you a picture, a picture of some of the people who are doing this. Well, if we start out with a form that is, let's say, two-dimensional, I'll be dealing with 2 dimensions. 3 dimensions, 3 dimensions gets a little hairier because of symmetries and trying to enforce them or deal with them somehow. 2 dimensions is a little easier. You can imagine a form that has landmarks. And a landmark would be something where it's a known reference point on the image. If you're looking at a sideways image of my face, it might be the tip of my teeth at the front. It might be the center of my ear. Something that is not just an arbitrary point along the edge of the form. I'll say that there are methods called semi-landmark methods for dealing with continuous curves. I'll mention those again once, but mostly we'll talk about landmarks. So you imagine that someone has taken this form, taken an image of it, found the reference points, and digitized them, and either done so by image processing or by using a digitizer, and got a series of XY points, and that all the point ones in the different forms are all held to be the same landmark. And I'll show you images in a moment that make it make it more concrete. Here there are P landmarks, so you have 2P coordinates x1 through yp. Okay. Well, you might say, okay, let's just use those. Why don't we, for a first cut, just say, take those as numerical scales and say they're quantitative characters. Let's go do Brownian or OU or something on a phylogeny and analyze them in the usual way. You know, just treat them as numbers. We don't have to think any more about the morphometrics. There's a problem, and the problem is to avoid meaningless translations or rotations of the specimens. And it can be thought of, as the problem of superimposing the specimens on each other. Although watch out, we're going to reject that in a moment. And the idea is also that if you have a fish that's somewhat two-dimensional, like a somewhat thin fish, and you're going to digitize landmarks around the edge of the fish, and somebody goes to do it, and they take the fish, and there's a digitizing surface. They will tilt it up and do it vertically, and they approach the surface, and they put the fish on the surface, and you'll notice that what they do is they make an arbitrary choice of exactly where to put it, here or here or here or here. Horizontal location becomes a matter of them choosing. Same with vertical, and same with rotation. They could rotate it 2 degrees, 5 degrees, and the question is, does that matter? That will affect all these coordinates. So if you just take them raw, you are accepting the decision of whoever was digitizing it as a biological property of the specimen, and that's not a good thing. Okay. Now, you might think it's a matter of superposition, but here's a problem with superposition. Here are two lineages evolving. This form here is a fish. It's got 13 landmarks, the points where the corners here. They're identical. If you might say, well, let's superpose in such a way. Here is this fish. It evolves. It becomes this fish. And what we see when we superpose them is that we can perfectly superpose all the front areas of the fish, but the back 4 landmarks have moved back 1 centimeter. Okay, over here we did the superposition differently. We superposed the tails. We started with the same form. We go up, we evolve, we get this form, and this form has the property that these 4 landmarks are the same, and these 9 landmarks, I forgot to draw the little 2 red arrows here, are moving forward 1 centimeter. Okay, question, are these this is a biological change, the tail moves backwards. This is a biological change, the front part of the fish moves forward. Are those different? Anybody have an opinion? Could it? Yeah, sorry.
It's irrelevant, right? I mean, because we don't care about where in the picture the thing shifted, but what the relative positions of one landmark, with respect to the other landmarks, right?
Okay, now he has opinion too, so I don't get both opinions out here.
Well, I guess from a biological standpoint, it depends. You have to look inside the fish to see what portion of the tail is elongating.
Right. But we only have the data from these 13 landmarks. Okay. We don't have the information on inside. Anybody else kicking in on that? There's another opinion. There's no candy to give out to the winner.
It's important to know...
There is candy to give out to the winner, but we're not sure. Yeah.
As drawn, they're probably not different. It's important to know how they differ when you center them on the centroid, right?
Well, we'll come to the centroid in a moment. But, okay, so there are three opinions. There's a fourth. A fifth also.
Oh,
yes.
sorry. Did I cut off before?
You're four. It's fine.
Is there, maybe there's some kind of a biological hierarchy of the landmarks? Biologically based hierarchy of which landmarks you think are important based on, you know, a priori
And would
information?
somehow tell you something about whether those are different.
Well, it would tell you which one to anchor it to.
Do you want to anchor to the most important ones? Something like that?
Maybe.
So you'd be allowing the most important ones to change the least. You'd be trying to achieve that. Okay, I mean, that's I see the view. You were entering the fray in hopes of getting a candy bar. Yeah.
I think the more the bodies gets progressively narrower towards the tail, the more difference these will be.
Okay.
Whether you stretch
All right,
tail or
so that's
move the
5 opinions now. Do we have any others? 6? 7? Sorry, we skipped you.
We were talking of underlying variables. I think you have to parameterize properly the growth of the differences or whatever to point to the reason for being different.
Well, I mean, these are all, you have the XY, I'm sorry. You have the XY coordinates of these 13. Are you saying you have to do something to those, or?
To make a model for measuring center of gravity or whatever. I mean, just...
Okay, well, we'll take that as another view, and you had your hand up earlier. So we're up to about 8 people. Oh, you have a microphone now? Okay.
Well, especially to reduce ambiguity, I mean, couldn't you define, I guess, where different landmarks begin and end, sort of break the whole silhouette up into several different polygons, and then measure the number of pixels contained within each polygon, and then compare those to each other.
Okay, those are certainly features you could derive from this. I mean, you could draw a line across here and count the area of that polygon. I'm going to shortcut the further discussion and show you the answer. No, they're not different. Why do I know that those aren't different? Because when I drew this figure, I drew this guy, then I picked it up and moved it over and dropped it down there. It's exactly the same form. I just located it such that the tail superimposed and the front didn't. But it's the same form. So it's got to be the case that this biological change, whatever it is the same thing as this biological change. So they look different. They're superimposed differently. One says, oh no, it's the front end that went forward, and the other says, no, it's the back end that went backwards. But those are the only thing you have. You don't have, somebody said, well, if that was a head, you'd have to ask where it was relative to the neck. And yes, that would be legitimate, but there's no neck here. There's no other piece of the form. So actually, the different superpositions here are your choice, not the fish's choice. And the forms are exactly the same. So in some sense, there is no correct superposition. This isn't more correct than that. Because what happened was that this part of the fish got 1 centimeter longer in both cases. And the fact that you might see it as the front going forward or you might see it as the back going backwards, that's your problem. You have to talk to your counselor about that, okay? Anyway, so you can, what they really mean is that the absolute locations of these don't matter. What matters is how far it is from this horizontally, from this landmark to that landmark and from this one to that one. The differences between horizontal differences or the vertical differences are meaningful, but the coordinate locations themselves are not meaningful. And some people touched on that. And there is a standard thing that they do in the morphometric consensus, which is the centroid was mentioned. If you take these points and take their average, that's the centroid of the specimen, and it is common, the usual procedure is to superpose them on the centroid. Now there's nothing magical about that. It's simply another arbitrary superposition, but it has the property then that it removes all arbitrary locations. You could also you could superpose them so that the point of the nose is exactly the same. And you could, well, we'll come to rotations in a second, but you could there's a thing you can do similar to rotations. So superpositions are arbitrary. The dimensionality of change of these things is smaller than you think. It's not 20 points. It's actually, in terms of location, it's actually no more than 18 numbers, because you have to remove the XY of just the positioning of the specimen on the surface. So what it amounts to is saying that what's meaningful horizontally is taking contrasts between points. Now, this is not the same thing as a phylogenetic contrast, because we're not talking about differences between specimens on a tree. We're talking about differences between points in one specimen. And so phylogenies don't get involved in it or anything. So you're centering so that the mean of the points are at 0, 0. That's a common way. It's not the only possible way. And you're dropping 2 dimensions, an x and a y, and now you have only 18 numbers, but there is the issue of rotation. Now, that former process of reducing the data down, in effect, what that does, reduce the data down to the horizontal differences and the vertical differences. And that is called, if you go through the mathematical terminology, that's called a Helmert transformation. There's a bunch of such ways of doing it, and they're all equivalently good, and they're all they're called Helmert transforms. Now we come to rotation. After all, the person putting the specimen down on the surface, they decided on some arbitrary angle at which they place the specimen. It's not a property of the fish, it's a property of the person. So here we have 2 forms, and one's a rotation of the other. I just picked up the same form and rotated it a bit. Now you'd hope that there was a similar transformation you could do on those forms that had the property that it removed, that it dropped 1 more degree of freedom, an angle of rotation, and ended up with a set of numbers that were nice as a result of that. The annoying feature of the mathematics is there isn't such a transform that I know of, so we have to do something else. What we're going to be doing is doing something that ends up with a likelihood analysis, or Remel likelihood analysis. And one way of handling rotations, the way we will use, is to associate with each specimen an angle and optimize take with the first specimen and just leave it where it was, and rotate the others with respect to the first specimen, and consider those as parameters of the problem and solve it by maximum likelihood, not by any cute trickery, but just by rotate a little, compute the likelihood, rotate a little, compute the likelihood, until you finally get them all rotated jointly. Okay, so we have the location and rotation things to take care of. Helmert takes care of it nicely, and you can show whichever Helmert method you use that they all end up with the same likelihoods. Wish I had a rotation scheme that was non-arbitrary, that acted like that for rotation, but we use this maximum likelihood thing. What do the people who do the morphometric consensus do? They have something called, they superpose specimens by what's called the Krustys transform. Here are two triangles. What you do to do it their way is you take the corresponding landmarks, you know which those are. This is landmark number 1, this is landmark number 1. You measure, you have a vector between them, a line between them, and you measure the fit of these figures to each other by summing the squares of those distances. So it's a least squares fit between two forms. And what you do is you achieve locations and rotations such that you minimize, in the case of the generalized Krustys analysis, GPA, you minimize the sum of squares. They also change sizes. They rescale sizes. The stuff I was talking about, we hadn't yet changed sizes. They also change sizes so as to minimize this. It has some nice analytical properties. If you superpose centroids, that takes care of location and is the minimum solution. And there's some for two forms, one against the other, there's an analytical calculation involving moments that flips them into the optimal, flips one of them into the optimal position with respect to the other rather easily. Now the algorithm that they use for multiple forms is just take all the forms, superpose each one using this generalized Krustys least squares rather quick method to the first form, numbers 2 through n. Take the averages of the coordinates to get an average form, then take that and superpose everybody to that. Then average again, and then go back and superpose everybody to the new average, and you keep going. This converges very rapidly. In about 3-4 cycles, you're down. You get it down to 8 decimal places or something. It goes really quickly, and it's fine. It's a fine way of superposing. It uses this least squares method. However, notice it doesn't use the phylogeny at all. It just treats the specimens as if they are IID. It also makes some implicit assumptions about statistical noise. Right now, this just seems to be geometry. Statistics is sort of not there. But the morphometric consensus starts with the generalized Krustys analysis, the GPA. And there were some lovely theorems done by two guys who did important work. The late David Kendall was a very important British probabilist. He did lots of important things. Late in his life, he got into this, and he came up with the Kendall shape space and these Krustys, which is related to these Krustys methods. And what they do is they superpose by a Krustys fit all the specimens. Then they compute all the pairwise distances between forms. You have this sum of squares thing for all pairs of forms. And I should say the generalized Krustys analysis, when you're doing it with multiple specimens, has the effect that what it is minimizing is the sum of squares of differences among all pairs of forms added up. It's kind of a joint measure of fit. And that process that I told you about with the mean, recomputing the means, does that. So you compute all pairwise distances between forms. And now you have this bunch of distances. And the interesting thing that the morphometric consensus does, at that point, it sets aside the data and just takes these distances. It computes a principal components for the distances. Now, if you think about it, principal components usually do by having a cloud of points in space and you put axes through them. But if you think, if you had a set of, and also if I take a cloud of points and put axes through them, if I take that whole cloud with the axes through it and I rotate it or I translate it, that shouldn't at all affect which axes I choose. And what you can show basically is it depends only on the distances between the points in space. So they use that, they use a set of distances, and there is a counterpart to principal components, which is in effect just using the distances between the points, getting the axes from that directly from the distances. And it's called principal coordinates, not components. And it was invented in the 1970s by this guy, John Gower, who I think is still alive. Then what they do is take those principal coordinates, axes, and analyze them. And you might say, this is a little weird. The specimen coordinates got lost and you have just these distances. What Kendall was able to do was to show that if you have measurement error such that each point is deviating from where what it should be by a normal distribution. So we have a specimen, we look at this point and we say, well, different measurements of the specimen will differ because there will be a circularly symmetrical isotropic bivariate normal distribution, meaning there will be error in X and error in Y, and they won't be correlated with each other. And this error will be the same at every point and all independent from each other. So it's a simple model of measurement error. You make an independent error left and right and up and down each time you digitize, and then the next thing you digitize, you make similar error but independently. And what Kendall was able to show, which really was a tour de force, was that if that's the model of statistical error, it is preserved in an appropriate way in this strange distance calculation, that the statistical noise is properly preserved, and so it's okay to go off into the Kendall shape space and the tangent space to that, which is another wrinkle. But it implicitly assumes that the model of error that you're dealing with when you're analyzing your specimens is independent. It's isotropic in the sense of the up-down variance and the left-right variance are the same, and the same in every specimen, in every landmark. And as long as that's your model of statistical error, the morphometric consensus is validated. It's okay. It's an okay way to do everything. Okay, so the problem that it has is what's called the Pinocchio effect. So here's Pinocchio. He just told a lie and his nose got longer. Okay. Now, if you're analyzing data from Pinocchio, this is evolutionary change, temporal change anyway. And the error, the change, the amount of difference between isn't just independent, circularly symmetrical fuzz at each point, at each landmark. It's much bigger in the tip of the nose, and it's much bigger left to right than it is up to down. So it's outside of Kendall's model. Here is what a Krustys fit might do to Pinocchio before and after. It would compromise the superposition of all these other points so as to get the nose more correct. Because, you know, with sums of squares, the largest difference starts to have a big, big effect. So a Krustys fit might do that to Pinocchio. What you'd like to do, of course, is that, and have Pinocchio tell us that, oh, look, the change is mostly in the nose and not elsewhere. Well, this issue has been faced by somebody else other than us at the same time. And that's Doug Theobald, who's a very good molecular phylogeny and molecular and protein structure-y person who was at Brandeis University. And what he was doing was superposing backbones of proteins. Here's a bunch of them superposed. Here they are superposed by least squares. What you'll notice is they have some parts that don't change much, and then they have a certain part where it's just sort of sticking out and wiggling around and not very well superposable. It doesn't really fit the Kendall model because the noise is much less there than down here. So he came up with the idea that the noise should be an arbitrary covariance matrix that you want to estimate. And then he came up with a maximum likelihood method of superposition. He made some more assumptions. And here's the superpositions he gets. He gets very good superpositions up here because when he gets down here, he just kind of lets it go to hell. He basically, his method will estimate that the covariances down there are big, the variances are big, and it will try less hard to superpose down there because it will say, well, we don't have as much penalty for not superposing because we know that's an area that's very fuzzy, that has a lot of change between molecules. By the way, he doesn't, you can't change size. He doesn't have any notion of, he doesn't use any notion of size here because you can't rescale a protein molecule. You can't change the number of angstroms that the two atoms are apart from each other without destroying chemistry and probably the universe. And he has a couple papers. This is his big paper, Theobald and Wuttke in 2008. So it does not assume isotropic independent error, but it estimates a full covariance. You might say, well, is he doing phylogenies? Doug is very phylogeny aware. He comes to evolution meetings. He's talked to me about contrast and stuff. But in the particular problem he's doing here, there is no consideration of phylogenies. It was not part of the problem. So he doesn't, but just because, not because he doesn't know about it. Okay. What we do, what Fred and I do, is very similar. We are computing likelihood of changes on a phylogeny using a covariant Brownian motion model, just the kind of thing we've been talking about. We're maximizing the likelihood using a given phylogeny. We're inferring the covariance matrix. That's what we've been doing. We choose the rotations iteratively to maximize the likelihood. So we have this whole analysis, and you take one fish and rotate it a little and do the whole analysis. But with computers, that's not too bad. And you keep doing it to get all the rotations decided by maximum likelihood, not by Krustys stuff. And we also incorporate size, what Fred calls scale. And I think I won't go into the details of it, but one variant we use is to infer a size character, scale everybody relative to it, and we do that inference by maximum likelihood also. But it's a separate size for each specimen. Okay, now when you look, and we're going down using the tree to compute the likelihoods, which if you're doing it PGLS, you could just do it directly, or you can do it by contrast. Same difference, won't make any difference which way you do it of those. Now, we did this with a superposition by superposing everybody on the centroid, which is what Krustys also does. This has the effect that any change along the tree will not be change of horizontal position or vertical position. It will only be change in other directions. So we can enforce that by making them superposing on the centroid. There are other ways we could superpose, and it would also lead to the same solution. Okay. So when you look through here and say, where's the morphometric consensus? We didn't do a Krustys fit to each other. We did put them on, superpose the centroids, same as Krustys. We handled the rotations differently. We didn't go off into we didn't compute distances and go off into a distance space and do our principal coordinates there. So really we dropped almost every step of the morphometric consensus, and we have an alternative to it. Centroid size, we can either do that or do this maximum likelihood thing for size. Centroid size, by the way, is the obvious thing. You take the centroid, you get the distances, the vectors out to all of the landmarks, including some, they can go outside the outline, that's okay. And you sum their squares and you square root that. So it's kind of it's going to end up as a root mean square of those distances. And that's the size calculated instead of estimated by maximum likelihood. And the morphometric consensus people scale everything relative to that size. Okay. There's also a linear approximation to size thing we do, which is close to the centroid size thing, but I'm going to skip it. Now you might say, well, how good is it to do coordinates, to assume Brownian motion of these coordinates? After all, they can do weird things like pass each other, and that's not so good. We're doing it, the Brownian motion approximation, on the implicit assumption that things haven't changed too much. They haven't changed enough that the points will get close and pass each other. And their justification for that, I think, is basically what used to be called, it's often called the delta method, which basically says that you can approximate, if you have a x that has a normal distribution, and there's a transform of x called z, some curve, that when you take all the x values and go over here, that if this variance is small enough, that thing will look more or less normal. Why? Because locally this curve looks almost linear. And when it is linear, this is normal means that's normal. When it's close to linear and there's not too much variation, it's still normal. Okay, so we may be working on the wrong scale for Brownian motion here, but as long as the changes aren't big, we can invoke the delta method and say, the variation, even if we're on the wrong scale, the variation is still normal, more or less. And that's what we are assuming. Let's see. I think all this I told you. Computing the likelihood, maximizing it with respect to the covariances, translations and rotations to maximize the likelihood. The translations, the centroid thing, does that. In effect, using contrasts, we have these two different size approaches, one ML and the other centroidy, and there's no morphometric consensus left. I think what happened was I duplicated this slide, didn't I, or something? Okay, I want to show you a simulation. Now, fortunately, you ran a case that was somewhat arbitrarily superposed fish forms from the simulation. Not a lot of you got it running, but you at least saw the figures, so maybe that can serve as that you've already done the lab. And basically what I did is I take this fish, which is the feared thresher salmon shark, and which is, fortunately, we're safe from it because it's hypothetical. And this fish has 10 coordinates, 10 landmarks, 20 coordinates. The change along the branches of the tree is like the measurement error. It's normal, it's Brownian wandering of each point independently. Those are shown by contour of the distribution, which is this green circle. Those are all independent of each other. So we're putting in a kind of noise that's very Kendall-like. But then, in addition to it, we're going to have the tip of this fin go up and down, wander up and down in a Brownian motion. And at this coordinate here, both coordinates are to change in a perfectly correlated way. So the tip of this fin goes out at a 45-degree angle. I think it's 45. So we have a covariance structure here that's not quite isotropic independent. It's got some correlated movements of fins in addition. Now let's see what happens when we do everything. We're going to assume a certain variance per unit branch length in the green stuff and a certain variance per unit branch length in the red position, going up and down. Here are, we have a tree of 100 tips. We simulate up it with that those known covariances. And then we center them and rotate them. They are different. Oh, I should also say, I didn't put it here, but there is a size change. There's another variable that's Brownian moving independently of these that is a log on a log size scale. Okay, I won't go too much more into that. And what you can see is, well, they kind of look like each other, but some things are changing more than others. Some are bigger than others. These are 20 of the a random 20 of the 100 fishes. When we rescale them by our maximum likelihood rescaling, there's what they are. They look, their sizes are taken care of, sort of. Now we take that and we just drop it in contrasts and get out principal components. Principal components. And then here are the same fish that I just showed you superposed. Now they're grayed out. If we were to, I believe what this is the morphometric consensus method. It gets a first principal component without the phylogeny. It gets the principal component, and it's kind of a little crazy. We know in truth that these two fins are doing a lot. Here, this fin is barely moving, and a lot of other things are moving too. And it's not really easy to see what's going on. The morphometric consensus has misinterpreted things. Why has it misinterpreted things? It doesn't have a phylogeny. It thinks those things are independent, identically distributed, so they're much more noisy. If we do the first shape PC with the tree, we get this, which shows, now the fins are moving a lot and the others aren't. But if you think about it, the centroids are supposed to stay the same. That means when the fins go up, everybody else has to move down a little in order to keep the centroid the same. So I came up with a way of dealing with that. I said, let's take now this is the first principal component, which means that everything is perfectly, when this movement occurs, these are not only perfectly correlated, this is perfectly correlated too. They're all perfectly correlated. Any lack of correlation is in the other principal components. And in terms of displaying it, I said, well, how about we take we take the median movement among all these vectors and subtract it out. That means you're no longer centered on the centroids, but you're moving the centroids of the fishes such that as much as possible of the movement goes into a few components. And when you do that, that's medianizing thing, there's what you get, which is now these guys are really little, and you can see that the motion is really where it should be. It's this fin motion that it's picking up. Let's see. Yeah. Actually, this is the morphometric consensus one, which is confusing. It gets some of the fin stuff right. I'm sorry, that first one wasn't. I don't actually know what the first one was. And you'll see sometimes I have the fin arrows pointing out and sometimes pointing in. You may say, oh, that matters. What matters is we want to know if they're perfectly correlated, are they moving in the same direction or in opposite directions? And the heads of the arrows indicate that. The heads could either be, you could turn every arrow around simultaneously and it means the same thing. Okay, so I'm sorry for the confusion here about the outcome of the simulation, but if you don't take the tree into account, you get more noise. That's clear. There are methods for so-called semi-landmark methods where you don't have landmarks, but you slide points around on the tips so as to optimize things, and we haven't really got that adapted to the phylogeny case yet. But Fred has many papers on semi-landmark methods, and we will get to that. There also are, I think, fitting forms by what's a very cute method called elliptical Fourier curves. We might try that at some point. All of this is somewhat geometric and does not explicitly take developmental or processes or genetic variation into account. In effect, the covariance of movements along the tree is incorporating, it's phenomenologically treating all the developmental and genetic stuff by just dealing with its outcome. Easy to say we should make use of all that. Of course, we mostly don't have enough information to do it. But it's important to keep in mind as goals that you really, in the long run, want to relate this kind of analyses to a knowledge of the developmental processes and a knowledge of what forms of genetic variation are available. If Evo Devo were doing comprehensive developmental analyses, then they could dream about connecting these two areas. Right now, Evo Devo is mostly concerned about a few genes of very large effect that affect shape and not all the other stuff, but they will get there. Our objective here, as with the other data, is the research program I mentioned earlier, which is comparing the directions of evolutionary divergence with the directions of available genetic variation to back calculate something about selective covariance, where selection is trying to push you. So references, this is the Morphometric Consensus, one of the volumes of that era. This is more complete statistical treatment, statistical shape analysis. Oh, you can't hear me anymore. Hang on, we'll see if I can recover. Dryden and Marty's volume is, very dense, and it has the theorems for the isotropic variation that justify the morphometric consensus. It's interesting when they get there, when they get to correlated things, they say, well, okay, what about the non-isotropic case? Let's deal with that. And they do some things, and they don't quite get anywhere, indicating they don't have any comparable machinery. There's Gower's paper. That's actually 1966. There has been a book, Geometric Morphometrics for Biologists, by Zeldich, Swiderski, and Sheets. I haven't looked at, that's the second edition of an earlier book. The earlier book didn't really have enough of the kind of thing I'm talking about. Maybe they've improved that in the 2012. So the methods that we have been hanging fire for over 5 years. I got to get that paper out the door and get the simulations finished. I hope to do so very soon. Anyway, if you look at this, a lot of the morphometrics comes in when you're superposing things and rotating things and scaling size. And once you get that done, the rest of the machinery is just the machinery that we know and love. So that's all for that. So that's a somewhat running over version of what I need to talk to you about. We could maybe take some time to discuss. This is the last lecture, so you'll be relaxing in a moment.
So, Professor, there are like some rather small group of species where there are actually rates of like cell division in different developmental stages. Could we use those rates to, I don't know, like scale or to weight the importance of each vector in your in that analysis to say, okay, it's more drastic if you move this part than if you move this part because of the developmental cell division rate?
Well, it would take the genes, whatever they're doing, are affecting those cell division rates. It would be of great interest to take forms at different life stages and do a joint analysis of their developmental paths and get lots of genetic principal components of that. It would be very, very interesting. It wouldn't necessarily solve the problem of what was easy to change and what was hard to change, because you'd still want to know how easy is it for a gene to change the cell division rate in different parts of the body. That would still be lacking there. But I think that kind of life history, or I forget, there's a phrase I'm missing, but that kind of developmental sequence would be very, very useful. Now, if slapping the fish down on the digitizing surface didn't kill it, you could do it. And I think in many cases you could. It'd be worth thinking about. But we're still, without the Evo-Devo-ish kind of stuff, we're still lacking an a priori way of saying what is easy what is easy to change in evolution and what is hard and what is correlated with what. And maybe someday the Evo-Devo will deliver that. Until then, we have to sort of empirically measure it. Steve, with microphone.
Back a few slides, you had a list of a slide called Geometry, Genetics, and Development. You said maybe the method should take, I guess maybe you think you should take developmental processes into account.
Well, to the extent that
What
you
did you
have
mean by that?
information on them.
Yeah.
You know what I think right now Evo-Devo is mostly talking about other things, and
it's all about gene expression. I too wish Evo-Devo would come to the party, but they show.
Well,
there are a few people interested in development that are trying to move.
Yeah, they're actually. You take some people like Sean Carroll.
Yeah.
There were a bunch of people in Evo-Devo who got up and said, "Oh, now we know about Evo-Devo, and we know that it's the right way to think about evolution. All these evolutionary biologists have been wasting their time. Evo-Devo is the right way to think about evolution," which I thought was very interesting, because it said if you took that seriously, you would be saying that, well, multicellular animals and plants have evolution, and the other single-celled ones, since they don't have a development, they don't evolve. Yeah. But Sean Carroll has been very good about, in spite of people dismissing all of evolution, he's gotten up and said, no, this is still inside the modern synthesis. Evolution is important. It's important to this. We have to think about it evolutionarily. It's just that it hasn't yet resulted in very much.
Yeah. So what I was going to say, though, is that this isn't your problem. I think it's a problem with the whole landscape approach. And that is that the landmarks you put down are chosen on the basis of repeatability and convenience and things like that.
They're like where sutures occur in the skull and
things
hit
that
the
you can
edge,
find on
things like
repeated, things that you can teach an undergraduate to repeat and so forth. It's not quite that simple. But the point is, if I were a developmental biologist, I might be a lot happier working with an X-ray of a flat fish, and then I would put my landmarks.
Well, they do.
Yeah. Okay. Well, but so the point is, you would use landmarks that actually represent underlying structures with like the chin is not really a character.
The chin is a sort of maximum point of curvature, and as you distort things, the corresponding point actually moves. The point that was the landmark no longer is the landmark. So the chin is a little marginal, or the tip of the nose. Those are marginal.
Right.
The corner of your eye is a little better.
But if we had developmentally informed choices of landmarks, that seems to me would take the whole system of landmarks to a place that would be a little more amenable to bringing development into.
Well, as I say, there are these analyses that don't use landmarks, and those could be used too. But I'm going to take a slightly contrary position to that. I'm going to say that actually you get enough landmarks, then whether you use those landmarks or whether you look more detail at the curves in between might not matter a whole heck of a lot, because you can kind of interpolate the one from the other closely enough that the differences are going to be kind of second order. Of course, if you have landmarks only in the front end of the fish and the whole back end has no landmarks, you're in trouble. But if you have enough landmarks, then you can either use landmark methods or non-landmark methods or hybrid methods, and I bet you get the same answers.
Okay, so
Yeah.
what if, of course, I'm not a, it's easy to talk about
Well,
these
neither
things
of
when
us
they're
is
outside your
actually
field.
doing this, yeah.
But do we still believe in the distinction between mesoderm and endoderm? Is that still,
I think
it's still
they
out there?
sort of do,
But
yeah.
What I'm saying is,
Last I heard.
if you could mark the boundary between those two fundamental tissues with different, with different fates and origins and so forth, that's what I mean by developmentally informed.
I think that will kind of start to happen as people make more use of things like CAT scans and also ultrasound collection of tissues. All of that is happening.